Okay, good evening, guys. I will share my screen, and one of you can let me know if I'm audible and if my screen is visible. Give me a minute. Sorry, 1 second. Okay. Can one of you let me know if my screen is visible and if I'm audible?
Yes, sir.
Okay. Thank you. Thank you. Okay, 1 second. Yeah. So, good evening, guys. My name is Aishi, and I'm a PhD student from Department of Chemistry, IIT Bombay. So, this is the seventh live session of this course, Fundamentals of Protein Chemistry. So, what we do in this live session, we basically discuss some of the old year assignment questions. So, we don't only solve the question, but we also try to understand the concept behind those questions so that it gets easier for us to solve similar type of other questions, right? So that is the basic purpose of these live sessions, and we want to keep these live sessions as interactive as possible. So I'll ask a question and I'll give you a few minutes to try to solve it, and then we'll discuss the concept of the question, the entire concept of the question. And if you have any doubts, you can unmute yourself and ask me or write in the chat box. If I know the answer at the moment, I will let you know. If I don't know the answer at the moment, then you have to give me some time. I will read about it, read much more about it, and let you know by the next session for sure. So that is how we'll go on with our live sessions. So with this we'll start with the first question for today. So the first question is, which of the following interactions is not directly responsible for the protein folding process? Option A, hydrophobic collapse. Option B, polar electrostatic interactions. Option C, hydrogen bonds, and option D, disulfide bonds. So you have to explain in detail which one is directly responsible and which one is not. So I'll give you a few minutes to answer. Anybody? Anybody? Okay, so the answer will be disulfide bonds. Okay, now we'll discuss. Disulfide bonds are definitely very important for protein folding and all of that, but they mainly stabilize a protein's final folded structure state. They are not the primary driving force that makes a polypeptide fold in the first place. So the main drivers of folding are the hydrophobic effect, the hydrogen bonding, and to a lesser extent electrostatic or polar interactions. So if you need a detailed explanation, hydrophobic collapse, basically this is directly responsible. Why? Because the hydrophobic effect is a dominant thermodynamic driving force for folding. Nonpolar side chains bury themselves away from water, which releases ordered water molecules and increases solvent entropy that produces a rapid compaction, that is a molten globule or collapse, that drives a chain towards native like conformations. Then we have hydrogen bonds. They are directly responsible. Backbone hydrogen bonds form alpha helices and beta sheets, the secondary structure, and are crucial for defining local structure. Side chain hydrogen bonds also specifies tertiary contacts and stabilize a folded state. Then we have polar or electrostatic interactions. They are directly and variably responsible. They are like charge-charge interactions, like salt bridges. Okay. Dipole-dipole and other polar contacts guide and stabilize long-range tertiary packing and can affect folding pathways and kinetics. So their strength is modulated by solvent screening, but they play an important role in many proteins. But disulfide bonds, they are stabilizing but not the main driver. Disulfide bonds are covalent crosslinks that lock in and stabilize a particular conformation once the correct cysteines are paired. They are especially important for secreted extracellular proteins, that is insulin, many secreted enzymes, where the environment is oxidizing. However, they usually form after or during late stages of folding and are not the primary thermodynamic cause of collapse. There are exceptions. For some proteins, disulfide formation is integrated into the folding pathway, like disulfide assisted folding, and is essential for correct folding. But that's the exception, not the general rule. Okay, the bottom line is the disulfide bonds because the generally stabilizing crosslinks rather than the main driving forces initiating protein folding. Okay. Okay. Now we'll not go into the basic things. We all know what is the chemical nature of amino acids, what are the groups in amino acids. We know the abbreviations and the side chain polarity of 20 amino acids, right? We have aspartic acid, glutamic acid, arginine, lysine, histidine, asparagine, glutamine, serine, threonine, tyrosine, alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan, etc. You can classify in a lot of ways, like polar amino acids, non-polar amino acids, some are aromatic amino acids, some are aliphatic amino acids, and all of that. So these are the basic amino acids because they have a basic side chain. This group is very basic because its positive charge is stabilized by resonance. If we talk about arginine, lysine is also basic, and for histidine, these nitrogens have a relatively weak affinity for an H plus and are only partly partly positive at neutral pH. These are the basic amino acids we know. These are the acidic amino acids, aspartic acid, glutamic acid, because their acid side chain negatively charged. Then we have the uncharged polar side chains. Polar side chains interact well with water, can form hydrogen bonds like asparagine and glutamine. They although the amide N is not charged at neutral pH, it is polar. Then we have serine, threonine, tyrosine. The OH group is polar, right? Then we have the non-polar side chains: alanine, valine, proline, phenylalanine, glycine, and all of that. Right? Leucine, isoleucine, methionine, tryptophan, cysteine. Non-polar groups energetically unfavorable for them to contact water. Then amino acids are linked together by covalent peptide bonds. Peptide bond formation with removal of water. Peptide bond in lysine and alanine, lysyl alanine. Okay. Now proteins are made up of a polypeptide backbone with attached side chains. Methionine, aspartic acid, leucine, tyrosine. You see, this is how peptide bonds are basically formed. Now, what are the characteristics of polypeptide synthesis? The amino group of one amino acid reacts with the carboxyl group of another to form a peptide bond, amide linkage, with the elimination of a water molecule. Now, the polarity of the molecule is retained. The monomers are all joined together in the same orientation. The polypeptide chain has polarity. The polypeptide contains a free amino group at one end and a free carboxyl group at the other. These ends of the polypeptide chain are referred to as the amino and the carboxy termini, or ends respectively. Now the product of the condensation reaction preserves the reactive group, carboxyl group, at the end of the molecule, and this makes it possible for the peptide chain to join to yet another amino acid. Now, what are the four levels of organization of protein structure? Like the primary structure of protein is obviously a sequence of amino acids linked together by peptide bonds, right? The covalent bonds, the amide linkage. Then we have the secondary structure of protein, polypeptide folding into alpha helix, beta sheet, or random coil. H bonds or hydrogen bonds are involved. Then the tertiary structure of proteins, 3D folding of a single polypeptide chain, H bonds, disulfide bonds, ionic bonds, van der Waals interactions, hydrophobic interactions, and all of these. Then you have the quaternary structure of proteins, association of two or more folded polypeptides to form a multivariate protein, bonds and interactions similar to tertiary structure. Then we have the unity and diversity of proteins. If you can see, now primary structure sequence of amino acids determines the structure of a protein, right? So, if you see, if there's a purified protein from isolated from cells, if it's exposed to a very high concentration of urea, we get the denatured protein. If we remove the urea, original conformation of protein reforms. In water, hydrophobic amino acid cluster inside a folded protein away from solvent. Why? So polar side chains, non-polar side chain, hydrophobic core region contains non-polar side chains and hydrogen bonds can be formed to the polar side chains on the outside of the molecule. Okay. So folded conformation in aqueous environment. Now secondary structure of proteins, alpha helix, H bonds between the NH of every peptide bond to the CO of the next peptide bond of the same chain. Now R groups are not involved. In protein, alpha keratin abundant in skin, hair, nails and horns and all of that. Okay. So secondary structure of proteins like beta sheet, polypeptide chains are held together by H bonds between the NH group of one polypeptide chain and CO group of the other chain. Okay. So in example in the protein fibroin, and which is abundant in silk. Now, alpha helices can wrap around one another by interactions between the hydrophobic side chains to form a stable coil structure, like alpha keratin in the skin and myosin in muscles. Now, tertiary structure of proteins, we talk about the 3D conformation or shape. Depends on the properties of the R groups of amino acid residues. They fold spontaneously or with the help of molecular chaperones. They are stabilized by covalent and non-covalent bonds. Now, many proteins are composed of separate functional domains. Example, bacterial catabolite protein. Protein domain are segment of a polypeptide chain that fold independently into a stable structure. Non-covalent bonds help protein folding. Also, if you see over here. Now, covalent disulfide bonds between adjacent cysteine side chains help stabilize a favored protein conformation. Okay, so they are not the direct factors involved in initiators or factors in protein folding. They help stabilize a protein conformation. So, like molecular chaperone proteins assist folding of other proteins. If you can see over here, these are basically chaperone proteins. If you see the schematic, unfolded peptide goes inside with the emergence of ADP and PA. This is a folded peptide that comes out. Now, how is the tertiary structure of polypeptide chain stabilized? Okay, they have ionic bonds usually between charged amino acid side chains at cellular pH 7. Okay, so hydrogen bonds between the R groups. Remember, uncharged polar amino acids can have H bond. Serine OH, threonine OH, and tyrosine OH. For example, uh, covalent bonds, disulfide bonds between cysteines. Now, hydrophobic interactions, non-polar side chains, associate with the interior of the molecule and exclude water, like van der Waals interaction. Now, the quaternary structure of proteins, hemoglobin, a protein in red blood cells has 4 subunits, 2 copies each of alpha and beta globins containing a heme molecule. Now, if you talk about gel electrophoresis, it's a method used to separate proteins. Okay. Now, so I'll not go into these details. So we know 20 amino acids are commonly found in the proteins are joined together by peptide bonds. A linear sequence of the linked amino acid contains the information necessary to generate a protein molecule with a unique three-dimensional shape. The complexity of protein structured in four organizational levels: primary, secondary, tertiary, and quaternary. Okay, this is the levels of protein organization. Primary protein structure is sequence of chain of amino acids. Secondary occurs when the sequence of amino acids are linked by hydrogen bonds. Tertiary occurs when certain attractions are present between the alpha helices and the beta pleated sheets. Quaternary protein structure is a protein consisting of more than one amino acid chain. Now, if we talk about the primary structure, it's defined as a sequence of amino acid proteins. Understanding the primary structure of proteins is very important because many genetic diseases result in proteins with abnormal amino acids that rises because of mutations, right? Which causes error in folding and a loss of impairment of normal function. Now, peptide bond. In protein, amino acids are joined covalently by peptide bonds, which are amide linkages between the alpha carboxyl group of one amino acid and the alpha amino group of another, right? Now, for example, the formation of the peptide between glycine and alanine is shown here. Peptide bond can be broken by prolonged exposure to strong acid and base at high temperature. To hydrolysis, these bonds can form bond enzymatically. So, if you can see, this is the peptide or amide synthesis schematic. Now, how to name the peptide bonds? If there is a free amino terminal to the left, that is the N terminal, and a free carboxy terminal to the right, that is the C terminal. And all amino acids are read from the N to C terminal end of the peptide. A polypeptide is linkage of many amino acids to peptide bond results in an unbranched shape. The residue or moiety each component amino acid in a polypeptide. So when a polypeptide is named, all residues have their INE or NIC or A changed to YL by exception of the C terminal amino acid. So what are the characteristics of the peptide bond? Partial double bond character, that is it's shorter than a single bond. Okay, because it has a partial double bond character. It's rigid and planar. This prevents free rotation around the bond between carbonyl and hydrogen of the peptide bond. Then transconfiguration because of the steric interference of the R groups when in the cis position. Uncharged but polar. Now, if we determine the composition amino acids of a polypeptide, the determining of the primary structure of polypeptide is to identify and quantitate its constituent amino acids. Okay, by hydrolyzing the strong acid at high temperature for 24 hours to release the individual amino acids which can be separated by cation exchange chromatography. If we talk about cation exchange chromatography, it's a mixture of amino acids bind to the column that contain a resin. It is a negatively charged group with different affinities depending on their charges and hydrophobicity. Separated amino acids contained in the eluate with solutions of increasing ionic strength and pH are quantitated by heating them with ninhydrin. It is a reagent that forms a purple compound most amino acids. The amount of amino acids is determined spectrophotometrically by measuring the amount of light absorbed by the anhydride derivatives. The analysis described is performed using a uh, amino acid analyzer. Okay. So next, now we'll skip this. We'll not talk about. So if you want to sequence the peptide from its N-terminal end, the beginning of the N-terminal end, phenyl thiazide known as the advanced reagent, and we have the advanced degradation process. It's a method of sequencing amino acids in a peptide. In this method, the amino terminal residue is labeled and cleaved from the peptide without disrupting the peptide bonds between other amino acid residues. The method can be employed by a machine called sequenator. Schematic diagram of the Edmond degradation. Now, many polypeptides are structure composed of more than 100 amino acids. These large molecules can be cleaved at specific sites, and the resulting fragments sequenced by using more than one cleaving agent, enzymes, or chemicals. Determination of protein's primary structure by DNA sequencing. The nucleotide sequence can be determined by knowledge of genetic code and translated into corresponding amino acid sequence of that polypeptide. This method has limitations of not being able to predict the positions of disulfide bonds in the folded chain and not identifying any amino acids that are modified after their incorporation to the polypeptide. Now, if we talk about secondary structure, it is defined as regular arrangements of amino acids that are located near to each other in a linear sequence. The alpha helix, beta sheet, and beta band are examples of secondary structure. So if we talk about the alpha helix, it's a spiral structure consisting of a tightly packed coiled polypeptide backbone core with the side chains of the amino acids extending outward from the central axis to avoid interfacial sterically with each other. The alpha helix we know is like a telephone cord shape held in place by hydrogen bonds between every NH group and the oxygen of the CO group in the next turn of the helix. 4 amino acids down the chain, very important. The typical alpha helix is about 11 amino acids long. Now, a very diverse group of protein contains alpha helix. Okay, the keratins are a family of closely fibrous proteins, major component of tissues such as hair and skin. Keratin myoglobin as globule, right? Flexible molecules whose structure is approximately 80% alpha helical, and the rigidity is determined by the number of disulfide bonds between the consequent polypeptide chain. If we talk about hydrogen bonds, it's an alpha helix is stabilized with extensive hydrogen bonding between the peptide bond and carbonyl oxygens and amide hydrogens that are part of the polypeptide backbone. So the first and the last peptide bond component are linked to each other through hydrogen bonds. Hydrogen bonds are weak, but they collectively serve to stabilize the helix. Amino acid potent will be each turn of an alpha helix contains 3.6 amino acids. Thus, amino acid residues spaced 3 or 4 apart in the primary sequence are close together when folded in the alpha helix, right? Now, what about the amino acids that disrupt an alpha helix? Okay, so if we talk about amino acids like that, so proline disrupts an alpha helix, right? Because its amino group is not geometrically compatible with the right-handed spiral of the alpha helix. Charged amino acids, example glutamate, aspartate, histidine, lysine, arginine, they disrupt the helix by forming ionic bonds or by electrostatically repelling each other. Then we have amino acids bulky side chains like tryptophan. Okay. Now, because they have a large number of amino acids, example like valine and isoleucine, they are branched at the beta carbon. Now, they can interfere with the formation of the alpha helix. Now, beta sheet is the second form of regular secondary structure in proteins. They are less common than alpha helix. The structure of beta sheet appear pleated. Beta sheets are often visualized as broad arrows. Now, comparison of the beta sheet and the alpha helix, if we do, beta sheets are composed of two or more peptide chains. Hydrogen bonds in beta sheet are perpendicular to the polypeptide backbone. Parallel and antiparallel sheets. Beta sheet can be formed from two or more separate polypeptide chains that are arranged either antiparallel or parallel. Antiparallel beta-beta sheet polypeptide chains are arranged to each other with the N-terminal and C-terminal ends of the beta strands. Parallel beta sheet street polypeptide chains are arranged to each other with the N-terminal of the beta strands. And the interchain bonds, the hydrogen bonds are formed between the polypeptide backbones of separate polypeptide chain. Now, interchain bonds is a beta sheet can also be formed by single polypeptide chain folding back on itself. In this case, the hydrogen bonds are interchain bonds. Now, beta bends or reverse turns, bends reverse direction of a polypeptide chain, helping it form a compact globular shape. They are usually found on the surface of protein molecules and often include charged residues. Beta bends are generally composed of four amino acid that cause a kink at the amino acids with the smallest R group. So beta bends were given this name because they often connect successive strands of antiparallel beta sheets. And beta bends are stabilized by the formation of hydrogen and anion bonds. Now, what is a non-repetitive secondary structure? Ends, groups and terms. So these are best recognizable as forms that resemble neither an alpha helix nor a beta-braided sheet. They do not have a repeating element in their amino acid sequence. These structures are often found on the surface of the protein. Now, what are super secondary structures or motifs? Supersecondary structures are usually produced by packing side chains from adjacent secondary structural elements close to each other. Example, alpha helix and beta sheets are adjacent in the amino acid sequence, are also usually adjacent in the final, like folded protein. Some common structural motifs combining alpha and beta sheets are these. So, what is a motif? A motif is an arrangement of a small number of secondary structures recurring in many different proteins. A motif is a short recurrence fragment of a protein, three-dimensional structure that is found in a large number of different proteins. A motif may contain two to perhaps seven or eight elements of secondary structures. The beta alpha beta motif consists of three secondary structures, the beta strand connected to an alpha helix connected to another beta strand. So motifs cannot exist on their own, they require the rest of the domain to maintain their conformation. If we talk about tertiary structure of proteins, tertiary structure is the total 3D conformation of an entire polypeptide chain including alpha helices, beta sheets and many other loops, turns and bends. The structure of the globular protein is aqueous solution, is compact with high density of the atoms in the core of the molecule. Hydrophobic side chains are buried in the interior, whereas hydrophilic groups are found on the surface of the molecule. This eliminates the possibility that water molecule may become bound to these hydrophilic groups and disrupt the integrity of the protein, right? So the alpha helix and beta sheet structures provide maximal hydrogen bonding for peptide bond components within the interior of the polypeptide involved in the hydrogen bonds. Now, what is structural domain? Domains are the fundamental, functional, and three-dimensional structural units of a polypeptide. So, the polypeptide chain contains two or more domains. The core of a domain is built from combination of motifs. Now, what are the interactions stabilizing tertiary structure? Disulfide bonds. Disulfide bond is a covalent linkage formed from the sulfhydryl group of each of the two cysteine residues to produce a cysteine residue, where a sulfide bond contributes to the stability of the three-dimensional shape of protein molecule and to prevent them from denature in the external environment. Hydrophobic interaction is amino acids with nonpolar side chains tend to be located in the interior of the polypeptide molecule where they associate with other hydrophobic amino acids. In contrast, amino acids with polar tend to be located on the surface in contact with the polar solid. Hydrogen bonds, amino acid side chains containing oxygen or nitrogen bound hydrogen can form hydrogen bonds with electron rich atoms such as oxygen or carbonyl or carboxyl group of a peptide bond. Formation of hydrogen bonds enhances the solubility of the protein. However, ionic interactions, negatively charged groups such as the carboxyl group in the side chain can interact with positively charged groups such as the amino acid in the side chain of polypeptide. Now, protein folding, which occurs within the cell in seconds to minutes, employs a shortcut through the maze of all the folding possibilities. Now, as a peptide folds, its amino acid side chains are attracted and repulsed according to chemical properties, right? So, the role of chaperones in protein folding, there's another question related to it, but let me explain chaperones a little bit here also. A specialized group of proteins named chaperones are required for the protein folding of many proteins, proper folding of many species of proteins, and also known as the heat shock proteins, interact with the polypeptide at various stages during the folding process. Some chaperones are important in keeping the protein unfolded until its synthesis is finished or act as catalysts by increasing the rates of the final stages in the folding process. Protect proteins as they fold so that their vulnerable exposed regions do not become tangled in unproductive encounters. Now, what is the quaternary structure of protein? Quaternary structure is the arrangement of multiple folded protein or coiling protein polycultures in a multi-subunit complex. Okay, so if we talk about the denaturation of protein, protein denaturation results in the unfolding and disorganization of the protein's secondary and tertiary structure, which are not accompanied by hydrolysis of peptide bonds. Denaturation may be reversible into its original structure when denaturing reagent is removed and natural protein are often precipitated from solution. The denaturation agent can be different types: heat, organic solvents, mechanical mixture, strong acid base, and detergents. Okay, and ions of heavy metals such as lead and mercury. Okay, as lead and mercury. Now, what is protein misfolding? Protein folding is a complex. One second. Okay, so protein folding is a complex trial and error process that can sometimes result in improperly folded molecules. These misfolded proteins are usually tagged and degraded within the cell. Deposits of these misfolded proteins are associated with a number of diseases, including amyloid disease. Now, what is amyloidosis? Fibrillar protein assemblies consisting of beta-pleated sheets. Accumulation of these spontaneously aggregating proteins called amyloids has been implicated in many degenerative diseases, particularly the neurodegenerative disorder Alzheimer's disease. Okay, so the dominant component of the amyloid plaque that accumulates in Alzheimer's disease is A beta. So this peptide, when aggregated in a beta-pleated sheet configuration, is neurotoxic. So you can see the relevance of these studies. The A beta amyloid that is deposited in the brain in Alzheimer's disease is derived by proteolytic cleavages from the larger amyloid precursor protein. Now, a key component of these tangled fibers is an abnormal form of the tau protein. Now, what is prion disease? The prion protein PRP has been strongly implicated as a causative agent of transmissible spongiform encephalopathy, including Creutzfeldt-Jakob disease in humans, Krabbe in sheep, and bovine spongiform encephalopathy in cattle, popularly called as the mad cow disease. Now, the prion protein tends to form, it's really the mad cow, not made cow. Okay, the protein prion protein tends to form insoluble aggregates of fibrils, and it is highly resistant to proteolytic degradation, similar to the amyloid found in some other diseases of the brain. Now, PRP is a host protein. The key to becoming infectious apparently lies in changes in the three-dimensional conformation of PRP. So it has been observed that the number of alpha helices present in non-infectious PRP are replaced by beta sheets in the infectious form. So in Alzheimer's disease, normal proteins after a normal chemical processing take on a unique conformational state that leads to the formation of a neurotoxic amyloid protein assemblies consisting of beta sheets. So in TACs, the infective agent is an altered version of a normal prime protein that act as template for converting normal protein to the pathogenic conformation. We will not go into these things. So basically, protein misfolding and chaperones. Proteins can fold and say fairly fast. Protein funnel is a good model, extending beyond nearest neighbor interaction. Molecular dynamic will sometimes do a better job. Now, this is the protein folding funnel, the energy landscape theory. With the latest model that only worry about the nearest neighbor interactions, hydrophilic and the hydrophobic interactions. Okay. Now, misfolding I've already talked. Most proteins can spontaneously refold. Primary sequence determines tertiary. Some proteins do not. Okay. Now, most proteins probably go through several stages on their way to a stable structure. Some need help, particularly in crowded environment of the cell. Chaperones are protein molecules that assist the proper folding of other proteins, often are heat shock proteins because they help cell with elevated temperature, which tend to cause proteins to misfold. Also involved in the newly synthesized proteins where a lot of hydrophobic groups haven't yet folded up properly. Some chaperone systems work as foldases. They support the folding of proteins in an ATP dependent manner. For example, the GROW-L and the GROW-S system. Other chaperones work as foldases. They bind folding intermediates to prevent their aggregation, no ATP required. Diseases such as Alzheimer's, Parkinson, and mad cow disease are associated with misfolded proteins. Now, when intermolecular contacts are significant, we need to modify the energy funnel. Okay, idealized case, assume that 50% of the contacts are formed. Then can either fold normally or it can react with another partially unfolded protein and fall down the aggregation funnel. Okay. Lysozyme is a well-studied example of misfolding. It's an abundant in number of secretions such as tears, saliva, human milk, and mucus, also in egg white. They basically damage bacterial cell walls to catalyzing forms amyloid fibers. No, Stanley Prusner has added prions to the list of well-known infectious agents, including bacteria, viruses, fungi, and parasites. Prions exist normally as innocuous cellular proteins. However, prions possess an innate capacity to convert their structures into highly stable formations that ultimately result in the formation of harmful particles, the causative agents of several deadly brain diseases of the dementia type in humans and animals. This is called PRPS, where SC stands for Strepy, the original prion disease where it was discovered in sheep. Now, PRPS is very stable, resistance to degradation where number of proteases, heating chemical interference. Prion diseases may be inherited, laterally transmitted, or occur simultaneously. Now, regions within diseased brains have a characteristic porous, spongy appearance, evidence of extensive nerve cell death, and affected individuals exhibit neurological symptoms including impaired muscle control, loss of mental acuity, memory loss, and insomnia. So, prion in the scaly form is an infectious agent composed of protein in a misfolded form. So, this is the central idea of the prion hypothesis. This would be in contrast to all other infectious agents like viruses, bacteria, fungi, or parasites, all of which must contain nucleic acids, either DNA or RNA or both, and PRPSC can catalyze PRP to amyloid fibers. Now, plants are not considered living organisms and are misfolded protein molecules which may propagate by transmitting a misfolded protein states. If a plant enters a healthy organism, it induces existing properly folded proteins to convert into the disease-associated misfolded plant form. So you see, it's contagious. The plant acts as a template to guide the misfolding of more proteins into protein form. Okay. So these newly formed plants can then go on to convert more proteins themselves, and this triggers a chain reaction that produces large amounts of the plant form. So all known plants induce the formation of an amyloid fold in which the protein polymerizes into an aggregate consisting of tightly packed beta sheets, right? So the figure this schematically illustrates how variants of disease causing plants affect different parts of the brain. So in bovine spongiform encephalopathy encephalitis, the brain stem is affected, right? In fatal familial insomnia, humans, a different mutation of the PRV protein, the thalamus region. In Creutzfeldt-Jakob disease, the cerebral cortex. In KURU, cannibalistic humans in Papua New Guinea. And in Gerstmann-Straussler syndicate, humans inherited from disease cerebellum is damaged. Now, protein amyloid aggregation has been recognized as a major cause of several important diseases, including Alzheimer's disease, Parkinson's type 2 or non-insulin dependent diabetes, and the transmissible spongiform encephalopathies. So, about 17 different proteins have been found to form amyloid in vivo. Amyloid fibrils formed from those proteins share some common morphological features, but these proteins do not have a conserved sequence or native structural motif. Now, protein amyloid fibers are often found to have a beta sheet structure regardless to their sequence, leading some to believe that it is follicles misfolding that leads to aggregation. There is a lower energy state in fibers. Okay. So enzymes basically act on the APP amyloid precursor protein and cut it into fragments of protein, one of which is called a beta amyloid, and it's crucial in the formation of senile plaques in Alzheimer's. Now, how proteins help in folding? The chaperones. So in eukaryotes, the proteins HSP6 and HSP10. Sorry, I have to adjust this. So in eukaryotes, the proteins HSP6 and HSP10 are structurally and functionally nearly identical to the Rho-EL and Rho-ES respectively. So in general, purely understood how it works. However, it is active in terms of interactions between chaperonins or inert, where chaperonins simply make an environment conducive to folding is not known. It's however required for the proper folding of many proteins. Okay. So what are the steps of chaperonin action? An unfolded polypeptide enters the cylinder from one end. The cap attaches, causing the cylinder to change its shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. The cap comes off and the properly folded protein is released. Chaperones. So with ATP, environmental conditions suitable for proper folding. Okay, this is the ATP dependent folding along a smooth energy landscape. Okay, so binding of 7 ATP molecules to the GRO-EL ring triggers conformational chain that results in a slight twist and tilt in the subunits and in exposure of hydrophobic patches that interact with and help to unfold misfolded proteins. Okay, so protein folding. So if you talk about Anfinson, if you know about Christian Anfinson, in case of Anfinson's experiments, the assumption was that amino acid sequence are completely and uniquely determines the protein's tertiary structure. If the protein folding problem is find native conformation among the large number of alternative conformations. Polypeptide chain of 100 residues can have 9 to the power 100 different conformations. Okay. So protein folding step by step is the extended chain, disordered globule, hydrophobic inside, hydrophilic outside, and then we have the native highly ordered conformation. So factors which influence the thermodynamics and kinetics of protein folding: size, amino acid content, hydrophobic or hydrophilic content, strength of intermolecular interactions, number of SS bonds, and domain architecture, right? So, what is the fold recognition? Unsolved problem, direct prediction of protein structure from the physicochemical principles. Now, solved problem is to recognize which of known folds are similar to the fold of unknown protein. Now, fold recognition is based on observations or assumptions. The overall number of different proteins folds is limited. The native protein structure in its ground state, minimum energy. Okay. We'll not go into homology modeling and these all of these things. We'll not go into molecular dynamics. So proteins are basically synthesized on ribosomes as linear polypeptides, right? And as they synthesize, they assume secondary and tertiary structure. The activity of the proteins depends on the integrity of its final tertiary structure, also referred to as a native form. Now, the native form of protein is not very stable structure and it's vulnerable by change by heat, high salt, reducing agents, detergents, or organic solvents. Now, all these treatments destabilize the weak interactions responsible for the higher order structures. Destruction of the higher order structure leads to the loss of activity of a protein. This process is referred to as denaturation or unfolding of the protein. Denaturation may not require complete unfolding of proteins. Okay, it might still be a folded structure, but in random conformation. So denaturation is cooperative. That is, changes in one part of the protein accelerate the unfolding of the other part. So some proteins are resistant. To denaturation by heat. The proteins of hot spring bacteria are stable at 100 degrees Celsius, and the primary structure of these proteins are not very well different from those that of the normal bacteria. So it remains a biochemical puzzle to explain the stability of these proteins. So heat stabilizes H bonding, detergents, urea, organic solvents, destabilize hydrophobic interactions. Extreme pH conditions cause ionization of the cytene, resulting in electrostatic repulsion and collapse of structures. Okay. Christian and Finson in denaturation of ribonuclease by urea and reducing agent led to complete loss of activity. Now, removing the urea and the reducing agent from this mixture resulted in complete denaturation of the enzyme. Okay, so 8 cysteine residues and 4 sulfide. Okay. So mathematically there could be 105 ways to make this bonds, but only one way of the native peptide is assumed after renaturation. Now chemical synthesized enzyme used in this study also shows similar results, ruling out any role of contaminants in renaturing process. This is a native state, renaturation of ribonuclease. Addition of urea and mercaptan unfolded state disulfide cross-links, reduced E-cysteine residues, removal of urea and mercaptan, native catalytically active state disulfide cross-links correctly reformed. Now, folding of polypeptide is rapid in a stepwise fashion. Proteins are synthesized and folded in different conformation, and the one of which matching with native conformation is selected. Trial and error method, not possible. The proteins keep folding in the correct conformation as it is synthesized in a stepwise fashion. Initially, the unfolded peptide has highest randomness, and as it keeps assuming more folded structure, the entropy keeps decreasing. Not all proteins fold spontaneously as they synthesize in the cells. Folding of many proteins is facilitated by a specialized class of proteins called chaperones. Okay. So I have talked about all of these. Let's not go into much more detail. Okay. Okay, so coming back to the question that we started with. So, which of the following interactions is not directly responsible for the protein folding process? So as I said, the answer will be disulfide bonds because they're generally stabilizing crosslinks rather than the main driving forces initiating the protein folding. So as I said, they are covalent crosslinks that lock in and stabilize a particular conformation once the correct cysteines are paired. They're especially important for secreted or extracellular proteins like insulin and many secreted enzymes, where the environment is oxidizing. However, they usually form after or during late stages of folding and are not the primary thermodynamic cause of collapse. Okay, so exception can be for some proteins disulfide pathway formation is integrated into the folding pathway, disulfide assisted folding, and is essential for correct folding. Right, so that's the exception of the general rule. So the answer will be disulfide bonds. If anybody has any questions, I'll wait for a few minutes and I'll move on to the next question for today. Anybody has any questions? Okay. See, there are no questions. I'll move on to the next question for today. Dash direct proteins along the correct folding pathway and protect them. What direct proteins along the correct folding path can protect them from other proteins that could impede the process? They can also inhibit misfolding and aggregation to some extent. Option A, enzymes. Option B, chaperones. Option C, cofactors. Option D, ligands. If you're answering, please answer in detail with an example as well. I'll wait for a few minutes. Okay, so the answer will be obviously chaperones. I already discussed in the last question, and also I've discussed many times about chaperones previously. So, chaperones are basically molecular chaperones are a special class of proteins that assist other proteins in folding correctly, right? So they don't dictate the final structure that's encoded in the primary sequence itself, but they guide, shield, and protect folding intermediates so that the protein can reach its native state without getting trapped in misfolded or aggregated states. So they protect exposed hydrophobic patches on a partially folded protein so that these don't stick to other unfolded proteins which would cause aggregation. So they are especially important under stress conditions like heat shock, oxidative stress, clouding, etc. when misfolding risk is high. So why not the other options? Enzymes generally catalyze chemical reactions, while there are folding related enzymes like protein disulfide isomerases and peptidyl prolyl isomerases. They are considered folding catalysts, not the general protective proteins that stop aggregation. Now, cofactors are non-protein molecules, metal ions, vitamins, heme groups required for protein function, not primarily for preventing misfolding. Now, ligands bind to proteins like substrates or signaling molecules, but they don't generally protect folding pathways. Okay. Now, if we want examples of chaperones, as I said, HSP family or heat shock protein 70 family binds to short hydrophobic stretches of nascent polypeptides coming off the ribosomes, prevents premature folding or aggregation until the whole protein chain is synthesized. Then we have the GROW-EL, GROW-ES system in bacteria. It's a GROW-EL is a barrel-like complex that encapsulates misfolded or partially folded proteins. GROW-ES acts as a cap, creating an isolated environment where the protein can fold without interference. So the proteins that protect other proteins along the correct folding path are B chaperones, such as HSP70 or the GROW-EL-GROW-ES complex, which prevent aggregation and assist proper folding. So that is the answer will be chaperones. So this is central dogma in molecular biology, right? DNA to RNA to protein. Now, great variety of conformations and shapes of proteins. We know it's a hugely diverse uh, thing that we have here, a very diverse set of proteins. Okay. Now, protein folding. How? Why does the protein latest follow conformation? So, under physiological conditions, the folding of proteins into the native conformation is spontaneous, right? Self-assembling process with no need of external templates to guide their formation. It was done by Anfinson thermodynamic hypothesis. The help of protein folding facilitating enzymes is often required, so mainly to prevent incomplete protein aggregation and to increase the speed of protein folding. Correct protein folding, right? Now, why is the protein folding taking place? Spontaneous, driven thermodynamically. Okay, because the folding obeys laws of thermodynamics to achieve the lowest energetic state for the given primary structure of the polypeptide chain under present physiological conditions to proceed in directions that increase the overall disorder or entropy of the system and its surroundings. The folding is accompanied by the reduction of free energy and or increase in entropy of the system. Unfolded is greater than folded plus energy, heat or entropy. So of protein primary structure it dictates protein's 3D structure. Now, living matter to attain the shape evolved by natural selection for the particular biological function. We have the Gibbs-Helmholtz equation. Delta G is delta H minus T delta S. So delta G is the used free energy, delta H is enthalpy, S is entropy. So if you have the delta G is basically the change in the free energy. It's a measure of the driving force of the chemical reaction. It depends on three parameters H, T and S. H is enthalpy change. It is the heat of the process that the process generates or releases, like exothermic or absorbs endothermic. So T is absolute temperature, obviously, and delta S will be entropy change, the chain conformational entropy change. So change in disorder. Now, if G is delta G is lesser than 0, the free energy of the product is smaller than the free energy of the reactant. Reaction can proceed spontaneously, it is exergonic. Now, if delta G is greater than 0, the free energy of the products is larger than the free energy of the reactants. So, the reaction cannot proceed spontaneously, it is endergonic, right? So, free energy plus protein folding, delta G is delta H system minus T delta S. Delta H will be bonding energies like disulfides, bound ions, non-bonding energies like electrostatic, hydrogen bonds, van der Waals, etc. What water and hydrophobic effect will be under delta S entropy. So the polypeptide is folded in three dimensions by non-bonding interactions between its individual atoms. Now, iron is A folding and denaturation. Physiological folding of proteins takes place in water environment. Now, if we talk about the hydrophobic effect, where two nonpolar side chains of phenylalanine residues come together, their surface area exposed to the solvent is reduced, and some of the ordered water molecules they have a lower degree of freedom in the phenylalanine solution shells are released to bulk solvent. The net disorder of the water molecules in the system is increased. So this increase in water entropy, that is greater than 0, is thermodynamically favorable and is the driving force causing non-polar moieties to come together, okay, in aqueous solvents. The proteins upon folding vary on average 85% of their non-polar side chains into their interiors. So this hydrophobic effect is the main driving force of protein folding. So the free energy and protein folding. So how do proteins build their native 3D conformation from their primary structure? Okay. Starting point with the peptide bonds, obviously two conformations, trans peptide bonds to groups with few exceptions. Assume the trans conformation, that is with successive C alpha atoms around opposite sides of the peptide bond joining them. This will be 10% of the peptide bonds. Now, peptide bond assumes a rigid planar structure because it has about 40% of the double bond character exists in two resonance isomeric states. Peptide bond length, no rotation allowed around the peptide CN bond. All atoms linked to C or N are in one plane. However, the backbone NC alpha single bonds can rotate like phi torsion angles. The backbone CC alpha single bonds can rotate side torsion angles. So the rotations are not unlimited. They are greatly restricted by collision of molecular orbitals of neighboring atoms of the polypeptide chain. Ramachandran diagram sterically allowed values of phi and psi. Now, what folding shapes or structures may a polypeptide chain attain that would be optimally compatible with the sterically allowed values of phi and psi? We have discussed about Ramachandran plot in one of the old classes, so we will not go into too many details. So, optimal folding shapes of secondary structure. So, regular secondary structure elements, alpha helix, beta sheet. Okay, in order to exist, the polypeptide chain conformation of angles phi and psi must not only fall within the allowed regions, but the particular conformation must also be stabilized. Okay, so in case of alpha helix, the glue that holds secondary structure elements is in part intrachain hydrogen bonds. Whereas in case of beta sheets, here are two stranded beta antiparallel beta sheets as known. Hydrogen bonding NHCO between neighboring polypeptide chains, interchain hydrogen bonds. Interchain hydrogen bonds. Turns or coiler loop conformation almost always occur at protein surfaces. Direction changes. Turns or coiler loop conformation almost always occur at protein surfaces. Direction change. So disordered regions, helices or sheets constitute 60% of the average protein. Now, non-covalent forces leading to protein folding into secondary structure elements and 3D conformation are contributing to protein's instability. So, non-covalent or weak interactions like electrostatic forces, ionic or ionic Stall bridges between charged groups, even though comparatively strong, their contribution to the stability of the proteins in aqueous solutions is low due to a high dielectric constant, okay, of water. So those on the surface of a protein do not usually interact with other charged groups of the protein, but instead are stabilized by hydrogen bonding and polar interactions with water molecules. Okay, we'll not go into too much details of these also hydrophobic interactions. So the formation of disulfide ores is not efficient in the cytoplasm due to its relatively reducing character. Okay, so they are formed in the endoplasmic reticulum or the ER, which is an oxidizing environment as the protein folds into its native conformation. So disulfide bonds give additional 3D structural stability to proteins described primarily for secretion into more oxidized hostile extracellular destinations with example uncontrolled temperatures or pH and before further protein processing or maturation. Example removal of a prosequent structure. If you have not known about, if you do not know about the levels and patterns, it's a very interesting concept basically. So, what is the way a protein takes to fold to its native conformation? Does the protein randomly explore all the available allowed conformations to it until it eventually stumbles upon its native conformation? So, calculations of sea level said that this cannot be the case. Assume that two n backbone torsional angles are there. Like 5 instead of an n residue protein, each have only 3 stable conformation. This is 3 to the power 2n, that is almost 10 to the power n possible conformations for the protein, which is a gross underestimate if only because the side chains are ignored. If a protein can explore new conformations at the rate at which single bonds can reorient, it can find 10 to the power 13 conformations per second, which is an overestimate. Okay. For a small protein of n, suppose 100 residues, the time t in seconds required to explore the conformations available to is 110 to the power 87 seconds. This is immensely more than the apparent age of the universe, which is 14 billion years, right? So proteins must fold in an ordered manner rather than by a random search of all their possible conformations. Each protein species appears to have evolved a conserved and simplified pathway of folding. So there's an energetic steps in the polypeptide folding. The folding follows an energy surface or landscape folding funnel that represents the energy states available to the polypeptide under prevailing conditions. Polypeptides fold by a series of conformational adjustments that reduce their free energy or increase entropy of the system until the native state is reached. 3D foldings, pathway steps, hydrophobic collapse. What we were talking about. The protein's hydrophobic groups coalesce so as to expel most of their surrounding water molecules. And the initial state is known as the molten globule state. Okay, and formation of local secondary elements. So smaller regions fold separately and more completely than larger regions. Intermediate folding events, the native like elements are thought to take the form of subdomains. Example 5 to 100,000 milliseconds cooperatively. Final folding events complex several seconds. So summary, a protein molecule is in aqueous environment assumes its native conformation largely in response to the tendency of its surrounding water structure to be maximally disoriented, maximally disordered. Sorry. So the tendency to maximize the entropy of the protein water soluble system is considered to be the main driving factor in protein building. This involves the hydrophobic effect in which proteins upon folding vary on average 85% of the non-polar side chains into their interiors. By this way, the native conformation of a protein reaches one of the lowest, gives free energy accessible to the sequence. Despite that, all the conformational stabilities of native proteins are low, making them easily susceptible to denaturation. By altering the balance of weak stabilizing forces, example by heat, chemicals, detergents, pH. This marginal stability of most proteins under physiological conditions is an essential property that is arisen through evolution of function to be flexible enough to carry out their physiological functions. Some proteins may have two or more different stable conformations, alternative folding, conformational diversity of proteins. There are certain regions may adapt either chemical, helical, sorry, either helical or beta sheet structure dependent on the context surrounding. So folding to a conformation seems to be the main selective expression against this folding. So what molecular chaperones do is that they function to prevent or reverse improper associations, aggregations of polypeptide chain regions, particularly in multi-domain and multi-subunit proteins. So they do so by binding to unfolded or aggregated polypeptide solvent exposed hydrophobic surfaces and subsequently releasing them in a manner that facilitates a proper folding or 4D assembly. So, pathological associations in vivo are promoted by the fact that folding takes place in the presence of extremely high concentrations of other macromolecules. Consequently, unfolded proteins in vivo have a great tendency to form both intramolecular and intermolecular aggregates. This condition is very carefully monitored by the organism, and if it gets out of control, it may trigger an overall shock reaction called unfolded protein response or UPR. So, cells apparatus for handling of unfolded and misfolded proteins, chaperones and proteasomes to catalyze removal of unfolded proteins by either denaturation or degradation. Okay, so basically, the unfolded protein response of the UPR is a very important mechanism performed by the cells. The capacity of the cells unfolded protein control systems was exceeded, so gene signaling pathways are activated by unfolding protein accumulation. To cope with this risky condition by activating an effective response. So escape from the cell's apparatus for handling an unfolded and misfolded proteins. So abnormalities in protein folding and assembly may be important mechanisms of diseases, okay, like conformational diseases like protein degradation due to the incorrect folding caused by mutations like cystic fibrosis, the most common recessive genetic disease in the Caucasian population. Then we have the defective CFTR, which is the cystic fibrosis transmembrane conductance regulator. It's a glycoprotein, which is basically the one that is problematic defect. The deletion of phenylalanine 508 in 70% of the cases. Then we have pathogenesis like deletion of phenyl by 508 results in an improper post-translational processing of oligosaccharide chains. In ER, improper conformation of CFTR is like UPR degradation by the proteolytic surveillance system or proteosomes. So, despite the fact that the mutated CFTR still retains some biological activity, so clinic is highly viscous mucus that blocks the small airways in the lungs. Okay. So we'll not go into the details. We have talked about prion disease. Okay. So I'll not go into the details. Uh, so chaperones direct proteins along the correct folding pathway and protect them from other proteins that could impede the process. They can also inhibit misfolding and aggregation to some extent. So the correct answer will be chaperones. You have any doubts? Any questions, anybody? have any questions, maybe we can stop the session here for today. We'll meet again next Monday. Same link, same time. Thank you. Thank you. Okay, so we'll stop the